Hi there, my name is Mina Alvarado. I am taking medical physiology online through the University of New England during the summer of 2022. The date of recording is June 29th, 2022. Here's my name and the date. In this presentation, I will be exploring the human physiology of chemoreception, known as the senses of smell and taste. Here, I will highlight the importance of communication within the sensory nervous system and within the detection and processing of chemical stimuli. So here is an overview of chemoreception. Smell and taste are two of the 5 special senses of the human body, and as previously mentioned, are forms of chemoreception. Chemoreceptors are specialized sensory receptors that are sensitive only to specific adequate stimuli. For chemoreceptors, they are activated when bound to chemical ligands. Like other types of sensory receptors, chemoreceptors transduce their respective stimulus, meaning they convert this form of energy into an intracellular signal. The signal passes along an action potential on a sensory neuron until it reaches the central nervous system, where the signal is integrated and processed. Here we have 2 images which provide a visual overview of some of the concepts related to olfaction and gustation, which we will discuss later in the presentation. So, olfaction is the sense of smell. It is believed to be one of the oldest, most primitive senses to help us adapt to our surroundings and survive. Olfactory receptors are phasic, meaning that they quickly adapt after first receiving an odor stimulus, and they ignore olfactory information which has already been analyzed. This way, olfactory receptors can focus on and receive new stimuli to process. Stem cells within the basal layer of olfactory epithelium are constantly dividing and creating new neurons, which is unique to this type of neuron. No other sense in the special senses has this feature, which is unique. Next, we have the olfactory pathways. Olfactory information is the only type that does not travel through the thalamus. Odor information enters through the nose, then travels along the first cranial nerve to the olfactory bulb in the olfactory cortex of the cerebrum. It is believed that because this part of the brain has functions involved in the regulation of memory and emotional response, it's believed that odors are deeply connected to memories and emotions. So that's why if you are just doing something and then you randomly get a smell, it can take you back to a certain time or place that you associate with some deeper emotion. So that's how that is theorized. Olfactory epithelium is found within the nasal cavity and contains olfactory sensory neurons. Axons from these neurons form the olfactory nerve, which synapses with secondary sensory neurons in the olfactory bulb. The secondary neurons originate in the olfactory bulb and extend through the olfactory tract to the olfactory cortex. Here we have detailed image representing olfactory epithelium and some of the anatomical structures that make up the olfactory pathway. For signal transduction, we have sensory neuron dendrites on the surface of olfactory epithelium that contains cilia, which lay within a layer of mucus. Before being able to bind to olfactory receptors on this cilia, chemical odorant molecules must dissolve and penetrate through this mucus. Olfactory receptors are linked to a special G protein, which, when activated, increases a cell's cyclic AMP, which then depolarizes the cell. If the receptor potential is strong enough to reach its threshold, an action potential travels from the sensory neuron axon to the olfactory bulb, therefore conducting the olfactory information to the brain. And here we have two different visual representations of the signal transduction pathway. Here's one that's a little bit more localized, looking at the cellular response in the cyclic AMP transduction cascade. And here we see the pathway through the olfactory bulb and then to the olfactory cortex. Now we move on to gustation, which is our sense of taste. Gustation, which is closely related to olfaction, consists of a combination of 5 sensations, including sweet, sour, salty, bitter, and umami. Most of those are kind of self-explanatory. Umami comes from a Japanese word, and the taste of umami is associated with the flavor of glutamates, which are a type of neurotransmitter. Each of the 5 taste sensations corresponds with a physiological process. For example, sour taste is associated with the presence of hydrogen ions, and saltiness is associated with sodium ions. Here's the gustatory pathway. The taste receptors are located on taste buds, which are found dispersed in certain arrangements on the surface of the tongue. Each taste bud contains anywhere from 50 to 150 taste receptor cells, and these taste receptor cells are found in other areas of the oral cavity, such as the palate. In order to be tasted, substances first dissolved in saliva, and then they interact with membrane proteins on the taste receptor cell. This form of communication activates a signal transduction cascade, which results in the release of chemical messengers to gustatory neurons. The axons of these neurons follow the cranial nerves 7, 9, and 10, and they synapse in the medulla. Sensory information then crosses through the thalamus and reaches the gustatory cortex, where it is processed and interpreted as a taste sensation. Here we have 2 images representing the gustatory pathway and the path which gustatory sensation information travels. Now we're going to be talking about gustation signal transduction. Taste receptor signal transduction involves different cellular processes that depend on the taste that is being detected. Sweet, bitter, and umami tastes involve G protein coupled reactions, and the salty and sour tastes are facilitated by ion channels. Taste buds on the tongue consist of 4 different types of cells that perform several different functions. Type 1 cells act as support cells. Type 2 and type 3 cells are taste receptors. And type 4 are thought to serve as precursors of taste receptor cells. Sweet, bitter, and umami tastes are detected by type 2 receptor cells, which express a G protein known as gustducin, which begins transduction pathways that result in the release of calcium into the cell. The presence of calcium generates a release of ATP, which signals neighboring receptor cells and sensory neurons. The sour taste transduction mechanism involves an increase in hydrogen ions and the release of serotonin, exciting the primary sensory neuron. It is not entirely understood which cells are involved in detecting and transducing the salt taste, but it is believed that sodium entering the cell catalyzes an action potential that is fired by the primary sensory neuron. So this concludes my presentation on chemoreception. I hope you found this presentation informative of the ways in which the senses of smell and taste are detected and processed within the sensory nervous system. Thanks for watching.